Hi, my name is Sara Paull, and I am the disease ecologist here at NEON. I'm excited to talk to you today about NEON's pathogen datasets. I'll start with a broad overview of the NEON products that have pathogen data associated with them. NEON does pathogen testing for mosquitoes, small mammals, and ticks. The design for each of these protocols allows for collection of information about diversity and abundance, and mosquito datasets are also collected in a way that allows mosquito phenology to be tracked. So I'll start by talking a little bit about our mosquito collections. Mosquito trapping is designed to effectively capture the phenology of mosquitoes, so that the number of traps and the frequency at which we sample are dictated by how many mosquitoes were captured in previous bouts. So we have a nice figure showing the off-season and on-season bouts that we do here, where off-season bouts are in red and the on-season bouts are in green. And when mosquitoes are active and being caught, we call that the on-season. We conduct a single bout of sampling every 2 weeks at the core sites and every 4 weeks at relocatable sites. A bout of on-season sampling consists of one overnight sampling event where we set up 10 carbon dioxide traps, and then we follow that up with a daytime sampling event in which the traps are reset to capture daytime active mosquitoes. And then once we've had 3 on-season bouts where we capture no mosquitoes, so that's these last 3 bouts here where you don't see any filled-in squares, the domain transitions to off-season sampling. And off-season sampling consists of setting traps weekly instead of every 2 weeks, but they're only set at the core sites, and we only set 3 traps. And it's only nighttime trapping. And so that's what allows us to capture the phenology, because as soon as we capture more mosquitoes, we transition back to this on-season sampling. And the latency for the mosquito data product is 30 days. All captured mosquitoes are then sent to an expert taxonomist for identification. And the key thing to understand about these data is that sometimes the identification data represents subsamples from the overall trap collection. So anytime more than 200 mosquitoes are trapped, a subsample that contains about 200 mosquitoes is selected, weighed, and identified so that the total numbers and the ratios of species can be back calculated as needed. And then at the very end of the season, we send instructions about which mosquitoes should be prioritized for testing and which should be archived or pinned. So the mosquito pathogens are really rare. So we end up testing them in pools of 10 to 40 female mosquitoes that are split by site and bout, and we use PCR for our testing. And NEON selects which ones to prioritize based on which ones are most likely to be vectors for humans. So these typically include Aedes aegypti and albopictus, Culex tarsalis or pipiens, and Aedes triceriatus. And then oftentimes we'll have leftover mosquitoes that we can test any of the Aedes or Culex species as well. And the tests that we perform begin with a broad screen, and this is currently at the genus level to see if the sample is positive or negative. And then any of the broad screens that test positive at the genus level are then tested for the appropriate list of virus-specific pathogens. And I've kind of highlighted in dark blue some of the ones that might be more recognizable for people, such as chikungunya, Eastern equine encephalitis virus, dengue virus, West Nile virus, Zika virus, and lacrosse virus. So this is just a summary of the number of records that exist for different types of mosquitoes and pathogen data by year. So as you can see, we have a really robust mosquito data set with hundreds of thousands of mosquitoes that have been collected. And we test a smaller subset of these for pathogens. So I selected West Nile virus since that's one that a lot of people have heard of in the United States. And you can see we do up to about 5,000, 5,500 flavivirus tests in a given year. And then of those, a much smaller percent are positive, and that ends up leading to anywhere from sort of 0 to 30 or so West Nile virus positive in the NEON data. So moving on to discuss the small mammal data products, we trap small mammals at 6 to 9 grids per site. 3 of these grids are designated as pathogen grids, so that means they get 3 nights of trapping each bout. And it also means that these are the grids from which we collect the samples that go on for pathogen testing. And then anywhere from 0 to 6 of the grids are diversity grids, and that means they only see 1 night of trapping per bout, but it gives us a better sense of the diversity of the small mammals at the sites. And the grids are trapped using a 10 by 10 grid with 10-meter spacing between traps, which gives 100 Sherman traps per grid. And at the core sites, we do 6 bouts per year, and the relocatables get 4 bouts per year. And then, of course, Alaska has a shorter trapping season with only 3. And the latency for this data product is 45 days. So samples are collected from chrysetids, murids, and dipodids, and they're collected during 3 bouts out of the year. And up until recently, we were doing hantavirus testing. So we would test up to 140 blood samples per site for hantaviruses using the ELISA method. But we were receiving some feedback that it might be preferable to test animals for tick-borne diseases. And this is because many of these tick-borne diseases are currently emerging. They occur across broad areas of the United States, and they provide better linkages between the tick and small mammal data sets. And so starting in the 2020 field season, we're going to be testing ear tissues for the pathogen that causes Lyme disease, Borrelia burgdorferi, and blood tissue for a wide variety of other tick-borne diseases. And the latency in this data product is 270 days. And this is just a summary again of the number of records that exist for small mammal captures. And you can see that we get anywhere from 10 to 20,000 in a given year. The number of hantavirus tests that we've done, so anywhere from maybe 600 or so up to about 3,500. And then the number of animals that have tested positive for hantavirus. So this is anywhere from 16 to 100. So last but not least is the tick collection. So ticks are collected at 6 plots at every site, and a plot consists of a 160-meter perimeter that is then sampled using the drag flag method to collect ticks. And the tick collection occurs every 3 weeks at sites that are designated as high intensity. So these are sites where they have collected more than 5 ticks in the last year, and otherwise they're sampled at low intensity, which is every 6 weeks. And they're sampled between green up and greened out at a site, so typically starting in March and April and ending in September or October. And then all of the ticks are sent to a professional identification laboratory where all of the adults and nymphs are identified and the larvae are all counted. And the latency for the dragging product is 30 days, and for the identification product is 300 days. After ticks are identified, the nymphs are sent to another laboratory for PCR-based testing, and we test up to 100 ticks per bout. And the pathogens for which they are tested are dependent on the genus of the tick. So Ixodes ticks are tested mostly for Borrelia species pathogens, as well as the pathogens that cause babesiosis, anaplasmosis, and ehrlichiosis. And then non-Ixodes ticks are tested for pathogens causing ehrlichiosis, tularemia, Rocky Mountain spotted fever, and anaplasmosis. And the latency for tick pathogen data product is 390 days. So note that in most years we capture about 1,500 to 5,000 ticks. We test anywhere from 500 to 2,000 for given pathogens, in this case Borrelia burgdorferi, the pathogen that causes Lyme disease. And then we may get anywhere from 10 to 100 positives or so. So NEON really has a lot of data to offer in terms of hosts, vectors, and a growing pathogen data set that it's pretty exciting to see.